Hi, my name is Delia Vera, and this is color vision theories. But before we talk about color vision theories, we need to talk about rods and cones. Rods, rods and cones are two different types of photoreceptors located in the retina of the eye. They're responsible for detecting light and transmitting signals to the brain. Rods are sensitive in low light conditions and are located in the peripheral, while cones are responsible for color vision and are responsive to light. Um, what is trichromatic theory? Well, firstly, it explains color. It begins at the level of cone photoreceptors in the retina. Humans possess three types of cones, each containing a different photopigment that is sensitive to particular wavelengths in their range. The three cones are S cones, L cones, and M cones. S cones are sensitive to short wavelengths or blue light. M cones respond to medium wavelengths or green light. And L cones respond to long wavelengths or red light. Each type of cone absorbs light within a specific part of the visible spectrum. Light enters the eye, activates these cones in varying degrees depending on its wavelength composition, allowing us to perceive color. An example of this is when a light stimulates both L cones and M cones strongly but perceives S cones minimally, so the color we see is yellow. The coding system allows us to perceive millions of colors from just three types of photoreceptors. Light is transformed into a pattern of neural signals through specialized biological filters. These signals are sent to bipolar cells and eventually ganglion cells. Next is the opponent process theory. Opponent process theory describes how color information is processed after it is initially encoded by the cone photoreceptors. This theory explains how color is interpreted through comparative neural mechanisms within the retinal ganglion cells. The on-center retinal ganglion cells respond to a bright spot on its center and also to a dark ring on their surround. Off-center ganglion cells respond to a dark spot on the center and a bright light on the surround. This creates contrast and high acuity vision. RGCs respond to color cones, red versus green, blue versus yellow, and black versus white. This basically means that a cell may be excited by red but inhibited by green in the center of the visual field, while excited by green but inhibited by red in the surround. RGCs specifically receive combined input from multiple cones and are made to respond differently depending on the contrast between them. Retinal ganglion cells and opponent coding. Red and green retinal ganglion cells may be excited by signals from L cones, a wavelength of red, but signals will inhibit M cones. These are medium wavelengths by green signals. This push and pull response leads to enhanced color contrast. This allows visual system to highlight the differences between adjacent wavelengths rather than raw values. Additionally, it also explains why certain colors are perceptually exclusive. This explains how we can't see something that's reddish greenish at the same time due to mutual inhibition in red-green um, opponent channels. RGCs exhibit spatial opponency organized into the center surround receptive fields. On-center activation is once the cells are excited when the light hits the center of the receptive fields and is inhibited when the light hits the surrounding area. And on-center cells are activated by light swinging in the center. Off-center activation, on the other hand, off-center cells are activated by light shining in the surround, and by applying this, the dual organization allows RGCs to detect not only color but also edges, gradients, and contrasts in a highly precise manner.